Hello, everyone. Welcome to another PDX Ducks Science Night. We're so happy that you could attend our first Science Night of 2021. The PDX Ducks are a dedicated board that strives to engage, support, and maintain connections with local ducks. My name is Ellie. I am the current PDX Ducks president, and just make sure that you're checking your emails and following us on social media just to keep up to date with everything that we're currently doing. Also, thank you again for continuing to show up virtually. I know that this is a very weird time and we're still here 1 year later, but we definitely miss engaging with you all in person, but this is the next best thing. So we are happy with all the support that you continue to show us. We will be taking questions. Please use the chat function to submit any questions for Dr. Walker that we'll go over later. We'll also be doing a drawing at the end for two $25 gift cards to the duck store using a random name generator. So just make sure to stay until the end to see if your name is called. And now I will pass it along to Sandy.
Great, thank you very much. Well, once again, thank you. We do appreciate your support of our Portland Science Nights, even when they're virtual. We had an excellent one back in October, so this is our second virtual one. And I am very pleased to be able to introduce our speaker for this evening. Dr. Ashley Walker is an assistant professor of human physiology at the University of Oregon. Her research seeks to understand why vascular health worsens with age and how this decline in vascular health leads to an increased risk for atherosclerosis, I'm sure I butchered that, and Alzheimer's disease. Dr. Walker's research is funded by the National Institutes of Health, the Alzheimer's Association, and the John Lewis Family Fund. She also teaches a course in the physiology of aging to undergraduate and graduate students at the university. Dr. Walker earned her BS in exercise and sports science from Oregon State University, and her PhD in integrative physiology from the University of Colorado Boulder. She performed postdoctoral fellowships at Cedars-Sinai Medical Center and the University of Utah before joining the faculty at University of Oregon in the fall of 2017. And with that, I will turn it over to Dr. Walker.
Make it. Okay, there we go.
Okay, well, thank you for that introduction. I'm excited to be speaking with you tonight. As was pointed out, I have some affiliations with a different university in Oregon, so please forgive me. I am actually from Oregon. I grew up in Corvallis, and so this is my hometown. So as was stated, I am part of the human physiology department here at the University of Oregon. And for any of you that aren't familiar with the human physiology department, we are the third largest undergraduate major on campus. And we have students from a variety of, who want to pursue a variety of healthcare careers mostly. So a lot of our students want to go on to go to medical school or physical therapy school or nursing school or something related to that in the future. And we also have about 15 associated research labs. And so I run one of those labs. We study aging and vascular physiology. And since this is an alumni talk, I figured I would start first with showing you where we do our research. So my lab is located in Pacific Hall, and no matter probably when you were at University of Oregon last, I bet the outside of Pacific Hall still looks the same as it did when you were here. But the inside of Pacific Hall was most of it newly renovated a few years ago. This renovation was supported by a generous donation from the Ford family. And so in 2018, I moved into one of these sparkly new research labs shown down here on the bottom right. We have now filled that lab with a lot of equipment special to our research, and even during these COVID times, we're pretty busy in our lab working on these ideas of vascular function and aging. And so I have a couple of pictures here of students at work in my lab. But you're not here to hear me talk about our work. You're here to hear me talk about the research and Alzheimer's disease. So let's get started by talking about Alzheimer's disease. So you likely know someone or have someone close to you that knows someone who has Alzheimer's disease. In fact, every 65 seconds, someone in the US is diagnosed with Alzheimer's disease. That means during this talk, there would be 30 new people just within the United States that were diagnosed with this disease. In fact, In 2019, Alzheimer's disease was the 6th leading cause of death. So Alzheimer's disease starts as a disruption in the ability to remember and to think, and it eventually leads to this recognized sign of the brain shrinking. This is very debilitating to the individuals who have the disease, but it's also very hard for the family and friends who watch their loved ones go through this disease, and also who are tasked with the caretaking responsibilities. And as the US population continues to grow older, we're going to have an increase in the healthcare burden and the financial burden of this disease. So finding out ways in which we could prevent or treat this disease are really important. And to figure out how to prevent or treat a disease, we have to first understand what is causing the disease. So with Alzheimer's disease, one of the things you might have heard about was these plaques. So these plaques are just clumps of protein that form in the brain, and they can impair the way that the brain cells are able to talk to each other. So your brain is made up of cells called neurons, and these neurons generate electrical signals that travel from one cell to another to transmit a message. You could think of this similar to telephone lines on a telephone pole. I know we all use cell phones now, but back in the day when we all were using our landlines, you can think about this was the way to send messages from one place to another. But what would happen if these telephone lines got filled with debris, such as what if a tree fell on these lines? Now that signal is having trouble getting from one place to another. And this is sort of what the plaques are doing in your brain. This debris is making it so those brain cells can't communicate with each other as well, or could even cause those brain cells to die. And this leads to that inability to make new memories or to learn new tasks. So this is great, right? We know what's causing Alzheimer's disease, so we'll just clear this up. We can get rid of the trees, get rid of those plaques, everything will go back to normal, it'll be okay. Well, no. The problem is that currently there is no cure for Alzheimer's disease, and most of the medications for the disease aren't working very well. And the problem is that we have all these drugs that make it to the point of clinical trials, and then they're failing. And these drugs are mostly targeting these plaques. There's a few drugs that are trying to make the brain cells talk to each other better, but for the most part, they're trying to target these plaques. And so the question is it really these plaques that we should be targeting with Alzheimer's disease? Or is there something else happening in the brain? And so one of the other things that you might have heard of with Alzheimer's disease are tangles. And so the classic theory of Alzheimer's disease is that you get the plaques first, and the plaques cause the tangles, and then the tangles are causing the impairment in those neurons, those brain cells. So what is a tangle? So tangles are just damaged proteins inside those neurons. And so you can think about this as a frayed wire. So rather than something happening on the outside of the wire, now we have proteins that are inside the cells that are becoming damaged, and they're actually clumping together too. So in both these cases, we have proteins that are aggregating. But in this case, you have those wires that are fraying. So this could be one other cause of Alzheimer's disease, and plaques and tangles often go together. But what I'm going to try to convince you of today is that this isn't everything that's happening with AD. I want to convince you that we really need to be focusing on why did that tree fall down in the first place? Was there a storm? Is somebody going around cutting down trees? And most importantly, is it the tree that's the problem? The storm blew down the tree perhaps, but did the storm cause other damage to the telephone wires that we're not seeing? So part of the problem with understanding what's happening with Alzheimer's disease is it happens in the brain. So you can think about this, if you have something wrong with your liver and you go to your doctor and your doctor's like, I really want to know what's happening to your liver cells, they might send you to have a biopsy, right? We don't biopsy people's brains though. So it's really hard to figure out what's happening inside of the brain of somebody who might have Alzheimer's disease. And that means what we do know about this disease is mostly from people when we study them after they've died and we've collected their brain for an autopsy. So I have a question for you. It's a little bit of a quiz. It's kind of like you're back in class today. I do a lot of Zoom polls with my class, so you're getting engaged here with the Zoom poll today. So my question for you is, for individuals who are diagnosed with Alzheimer's disease, so they're diagnosed while they're alive, probably by tests of memory, looking at their risk factors, looking at the progression of the disease, After they died, in this study, they looked at their brains after the autopsy. I want to know how many of those people diagnosed with Alzheimer's disease do you think had plaques and tangles inside their brain? And so you have a choice of A, B, or C, which are 45%, 65%, or 85%. I just put polls in here because it's my chance to drink some water. Okay, interesting. So I'm going to actually share these results with you so you can see. I stumped you all because you're all over the place, right? Nobody really knows, which is sort of like we don't really know a lot about Alzheimer's disease either. So the correct answer was actually C. So a lot of these people do actually have plaques and tangles in their brain. So you could say that 85% of these people we probably diagnosed correctly when they're alive. So that's a good thing. That 15% maybe were misdiagnosed. And so if they ended up in a clinical trial for Alzheimer's disease, maybe that's why those drugs weren't working so well. But 85%, that's kind of good. But here's the next question. Is of those individuals, again, same people with Alzheimer's disease, when we look in their brains, what percent of them have something else wrong with their brain? So what percent of them have something, say, some other sign of neurodegenerative diseases or something wrong with their vasculature in their brain? So another poll, let me launch it for you. This is like the best of engagement I can do. I'm sorry, on Zoom. We're limited. We'd be shouting this out in the pub if we were there. Okay, so let me end this and I'll share it with you. And I guess my foreshadowing has done a really good job because a lot of people guessed C, which is 96%, which was the correct answer. So 96% of the people have something else going on with their brain. So this is actually the full kind of pie chart of what I'm talking about. And so in this chart, the purple slice is the people who just had plaques and tangles. So that was 3% of people. And then in the blue, which is most of the chart, is 82% of people, which are they have plaques and tangles and something else going on. And then in the orange was people who had something else going on. They didn't have any plaques and tangles, which was 14% of people. And then who knows about that 1% of people who had nothing going on with their brain and how they were diagnosed with Alzheimer's disease while they're alive. So since we're talking about brains, I want to talk about like what is the brain with you for a second. So this is your moment for a bit of relaxation. So we ask you to close your eyes for a second. Picture in your mind, what does a brain look like? Think about like what a human brain looks like. Okay, you can open your eyes, and I'm guessing you might have thought of something that maybe looked like this. Something that's kind of pink and wrinkly, right? The thing about it is if you see a brain like this, especially if it's a real human brain, what happens, when they do an autopsy on somebody is they clear out all the blood. And so if you had a human brain that still had all of the blood vessels on the outside, it would be covered in these blood vessels that you don't see when you normally think of a brain. And then if you dive deeper into that brain, you can see there's an intricate network of blood vessels within this. And so this is a picture from a plasticized version of a brain. So it was an actual real human brain that they injected with after the person died, obviously, with something that plasticized all the arteries, and then they melted away all the other parts of the brain. So what's left is this network of blood vessels. And this is really important, right? Because blood vessels carry the blood to the brain, and you need that oxygen, you need that glucose, you need the blood to take away the bad toxins in your brain. And so this intricate network is really important. And if it goes wrong, we're thinking that that could cause some of the things they're having with Alzheimer's disease. So to show you on this another slide, I have taken this pie chart and now changed it. And so now I still have in purple that 3% of people that had just plaques and tangles in their brain. But now I'm showing you in pink all the people who had something vascular related in their brain. So either they had little microbleeds in their brains or their vessel walls were getting thicker. And so that's 82% of people who had some kind of vascular damage and plaques and tangles, and then 5% had just vascular damage. And then we still have those people who had something else going on with their brain. So another way to think about this is let's consider what are the risk factors for Alzheimer's disease. So what are the common reasons, risk factors that would make you more likely to get Alzheimer's disease? First one is aging. So we know as you get older, you have an increased risk of getting this disease. But one of the other major risk factors is cardiovascular disease. And while cardiovascular disease can sometimes be about the heart, usually it's about the vasculature. So usually it's about those blood vessels. And so one of the strongest predictors of Alzheimer's disease is blood pressure. And this is where my interest in blood vessels and aging comes into play, because I know a lot about blood vessels and aging. And now I'm trying to figure out how is that related to Alzheimer's disease. So let's talk about what happens to your blood vessels as you get older. So with age, one of the main things that happens to your blood vessels is they become more stiff or they lose their elasticity. And just to orient you, I'm going to say this is happening primarily to your large arteries. And so your large arteries are your aorta, which is the artery coming off your heart, and the carotid arteries, which are the two ones in your neck that are supplying most of the blood to your brain. So those are the two arteries that are losing their elasticity with age. Your arteries aren't the only thing that lose elasticity with age. So you can humor me for a second. We're going to take our hands and we're going to pinch the skin on our hands. So if you pinch it up and let it go, you can watch how quickly does your skin bounce back to where it was. Now, if we were all in the pub together, we could look around at everybody else's table and see how long was it taking their skin to bounce back. And at this point, you might be thinking it's fortunate that we aren't in the pub, so you can't look at other people's skin and they aren't looking at yours. But if we were in the pub together, what you would see is people who are relatively younger, their skin is bouncing back quicker, and people who are older, it's taking longer for their skin to bounce back. And that's partly because there's elastic proteins within your skin, and there's elastic proteins within your artery that as you get older aren't being as elastic as they used to be. And we joke sometimes that this might be why grandmothers like to pinch their grandchildren's cheeks, because they just want to check and see, are those grandchildren still young and have that nice elastic skin? So much like your skin, your arteries don't spring back as well as you get older. And because your arteries are under really high pressure, unlike your skin, which is sort of loose, your arteries become really stiff to overcompensate for this loss of elasticity. And why this matters is because the blood flow through your arteries is not continuous, because the blood coming out of your heart is not continuous. So your heart is the pump. It's the pump that's pushing the blood out, and it's pumping at a pulsatile manner, right? So each time your heart pumps, it sends a big pulse of blood into your aorta. And so your aorta has a time of high pressure, and that pulse of blood is there, and a time of low pressure after the heart relaxes. And you probably know about this high pressure and low pressure because every time you go to your doctor, right, they take your blood pressure. So let's talk about what is blood pressure. So your blood pressure is made up of two numbers. Your doctor probably tells you you have a blood pressure of something like 120/80. And so these two numbers mean what the pressure is at the highest point in your arteries. So that would be your systolic blood pressure. That's the top number, like the 120. And what it is during the lowest pressure in your arteries. So that's the diastolic blood pressure. In this case, it would be 80. And so next question, I have another poll for you. This is the last one, I promise. No more quizzes after this. So what happens to your blood pressure as you get older? So the options are A, which is both your systolic and diastolic go up, so the top and the bottom number are going up. B is the top number is going up and the bottom number is going down. And C is the top number is going down and the bottom number is coming up. So as you get older, what is happening? Okay, I'm going to assume about everybody has chosen, and I'll share you the results so you can see them. And so this is typically what I get when I take this poll. I take this poll in my classes, I take it when I give talks, and usually the majority of the people say A, which is your systolic blood pressure goes up and your diastolic blood pressure goes up, right? Because we always talk about how aging is bad and blood pressure being high is bad. So blood pressure, of course, is going up for both. The correct answer is actually B. Sorry, let me exit out of the poll for a second. Okay. The correct answer is actually B. So your systolic blood pressure goes up and your diastolic goes down. And I can show you this on a figure here. And so on this figure, we have age, which is on the x-axis, so going from 30 to 80 years. And then we have blood pressure on the vertical axis. And so there's two solid lines, which are the important lines on here. So the top solid line would be systolic blood pressure, that's SBP. And as you can see, systolic blood pressure stays about the same until you hit your 50s, and it starts increasing, and then it increases at a higher rate as you get older. And then the other solid line on the bottom is diastolic blood pressure, that low number, and that actually increases through middle age, and then it actually starts to decline as we get older. And so the difference between your systolic and your diastolic blood pressure is known as your pulse pressure. And so we say that pulse pressure increases as you get older. So I know you're all wondering at this point, why am I talking about blood pressure in the heart? And you really came here to talk about brain and Alzheimer's disease. So before you like sign off your Zoom or start watching Netflix or whatever else you're going to do tonight, let me get back to why this is important to the brain. And that's because this increase in pulse could potentially be damaging to the arteries in your brain. And why this increase in pulse pressure is happening is because directly related to that large artery stiffening. And so on these two graphs here, I have on the left a young compliant artery. And so you can see the arrows are supposed to be the pulse coming in. So like this is going into, say, your carotid artery, so the main artery in your neck. So there's a pulse coming in, and each time that pulse comes in, that artery expands. It takes up sort of that pulse and slowly comes back and recoils. And coming out the other side is more of a continuous flow, less pulsatile. That's a little extreme. It's not exactly like that, but it's closer to that. And then on the right is the stiff or the aged artery, where you're sending a pulse in and that pulse is coming out exactly the other end. So we can think of the left healthy artery as the balloon perhaps, and the right as the straw. And so if we were in the pub together, I would be getting out the balloons and straws right now. And I'm sorry that you're going to miss that. So I made a video of it, which is not going to be as fun, but you can try this at home if you have a balloon and a straw. So what this video is of me, I took this last night, blowing into a straw in sort of pulses, or blowing into a balloon in pulses. And the balloon has a small hole cut in the end of it, so that there's air that can come out the other side. And so the amount of air going in the pulse is about the same between these two. And then the green piece of tissue paper is supposed to show you what the air coming out the other side is looking like. So on the top, when you have the stiff artery, we have the pulse coming in is about the same as the pulse coming out the other side. And you can see that tissue paper flaps in a pulsatile manner. On the bottom, with the compliant arteries, you see that the balloon is expanding each time I breathe into it, right? But that tissue paper isn't moving. It's moving a little bit, but it's not pulsing like it is below. So this is sort of like the arteries, right? Stiffer arteries mean that there's more pulse traveling up into the brain, whereas compliant arteries means there's less pulse traveling into the brain. And I'm just gonna give a little shout out to my daughter who I made hold this tissue paper, and she kept complaining about her arms being tired. And I'm like, no, this is for science. You gotta do this. But anyway, so moving on though. So what we think is that this more pulsatile pressure in your brain is causing or potentially related to Alzheimer's disease, because that pulse is causing the cells in your brain to become damaged. And so there's been numerous studies in human patients where they found correlations between stiff arteries, more pulse in brain arteries, and how much impairment there is in memory, or how much increased amyloid plaques there are in the brain. There was even one recent study where they had people enrolled when they had mild cognitive impairment. So mild cognitive impairment is the step before you get to Alzheimer's disease. So they had mild cognitive impairment, they enrolled them, they took their large artery stiffness measurements at baseline, and then they followed them for 5 years. And after 5 years, they saw that the people with the stiffer large arteries were more likely to develop Alzheimer's disease than the people who had the more compliant arteries. This means that stiff large arteries is predictive of future disease. So this is a major risk factor for getting disease. So in humans, we're able to do lots of studies with patients and make correlations. One of the things I'm interested in, though, is to figure out what is happening in the brain. Why is it that this greater pulse pressure is a bad thing? And what is it doing to the cells in the brain? And so to do these studies, we are actually using mice. The thing about using mice is that we can genetically change these mice to make their arteries stiff. And so I have mice that I've been able to make have stiff arteries when they're young. So I can have young mice with compliant arteries and then young mice with old arteries, essentially, and compare them. And so now I no longer have to worry about all the other things that happen with age. I can just look at what's happening if you have stiff arteries. So one of the first things we're interested in was looking at the blood flow in the brain. As I told you, these arteries in the brain are really important for getting the oxygen and the glucose to those brain cells when they need it. And so we're interested in knowing if you have stiff, large arteries, what does that do to the blood flow in the brain? So one of the things about studying mice is you need lots of specialized equipment, and we don't have all that equipment at the University of Oregon campus, but luckily we have some great collaborations with OHSU. And so up at OHSU, they actually have a machine that allows us to look at blood flow in the brains of our mice. And so we do this with an MRI. So an MRI is similar to an MRI that you would go to your doctor to get. You know, they talk about it as that claustrophobic tube with all the loud noises, right? To look at a mouse though, because they're so small, you need a really strong MRI. So a normal MRI, I think, is a 3 Tesla MRI at your doctor's office. This is a 12 Tesla MRI that they have up at OHSU. And it's small, so it just fits, you know, little rodents. But we're able to use this to look at blood flow. And I'd like to mention a couple of the people who worked on this project with us. So up at OHSU, Marty Pike is the one who is able to run the MRI. He figured out all the protocols for us and actually did the studies in the MRI with us. And then Nabil Elkyad is a collaborator who's helped us really establish our mice up there, transfer mice between the universities. And we're working on some other great projects together too. And then two people from my lab who did a lot of work on these studies are Grant Hinson, who is a laboratory manager. And he's the one who drove up to Portland repeatedly to do these studies. I think we sent him up there like for 3-day periods at like 5 different times. And so he was a trooper to drive between Eugene and Portland that many times. And then Nick Winder is a graduate student in my lab who did all of the analysis of the MRIs after they're collected. So this is what these little MRIs look like of this mouse brain. And so in these pictures, this is like a cross section of a mouse brain, and it's showing you the blood flow. And so dark colors, dark blue, bluish colors are low blood flow. And then the orange and the red is high blood flow. And so I've pointed with some arrows to some points that are really different between these two. So on the left, I have the healthy mice with the nice young arteries. And on the right, I have the mice with those old arteries. So they're young mice, but with old arteries. And as you can see, the healthy artery mouse has a lot more blood flow, so a lot more yellow and orange in their brain, compared to the stiff artery mouse, which has a lot more of the blue color in those regions. And then I have the graph on the right, so you can see that they're actually different between the two. So the stiffer arteries led to less blood flow. Next, we're really interested in wanting to know why this is happening. And so we can do things in my lab, like we can take the arteries out of the brain and we can study them in isolation. We can look at how well they dilate, how well they constrict, put all kinds of things on them and see how that affects the health of the arteries. What we also like to do is look at what the cells in these arteries look like, and as well as what kind of damage they have. And so one of the things we look at commonly is oxidative damage. So oxidative stress is a word that you've probably heard before. This is, sort of those free radicals. So reactive oxygen molecules that have, say, unpaired electrons, and they go around and damage other molecules. So they might want to steal electrons from other molecules. If you took chemistry, you might be remembering back to those chemistry days. They might want to steal electrons, or they might be going around damaging proteins or lipids in your body. And so your body needs these free radicals around. So I like to think about this as, say, rain. So we live in Oregon, right? We need to have some rain. So having a little bit of rain is good. Just like having a little bit of free radicals is good because they're signaling molecules. They're important to actually have like healthy homeostasis in your body. But you don't want to have really heavy rain, right? Because if it rains a lot, that could be damaging. You could have floods, or, you know, at the extreme, like say if it's hailing, then obviously this hail is going to be damaging things. And so what we think about this is that when we look at these arteries, one of them has a healthy amount of free radicals and one of them has too much. And so these pictures that I'm showing you are, again, the healthy arteries on the left and the stiff arteries on the right. And the red staining is staining for the amount of oxidative or free radical damage to these arteries. And as you can see, there's a lot more red in this artery on the right, showing that they have a lot more damage. And you might be wondering why these arteries look weird, like little figure eights. And that's just because these arteries have been taken out of the brain and they no longer have blood in them. So in the brain, they would have been nice little circles, but they're like smashed down on each other because they don't have any pressure and they're pretty flexible. So the thing to remember about this is that we didn't change the arteries in the brain. Our mouse has stiff, large arteries. So we changed the arteries outside of the brain. So we changed the carotid arteries and the aorta is stiffer in these mice. But their arteries in their brain are not stiffer. And one of the things that I'll point out in these pictures to support this is that on the figures on the bottom, you can see that green squiggle, looks like a neon sign, green squiggle. That green squiggle is actually the elastin protein. So it's the elastic balloon protein of these arteries, and it's the same between these two. So they have the same amount of elasticity between these two. So we really made the stiffness in the large arteries, but not in the brain. And so we're really showing that what's happening in the brain is really dependent upon what's happening to those other arteries. And so to summarize what we've seen with our studies so far is that we found that large artery stiffness causes lower brain blood flow and that it increases oxidative damage in brain arteries. And we've done many other studies to show that these arteries in the brain are unhealthy when you have more large artery stiffness. We also do things like look at their memory and their learning, and we can see that these mice have poor memory. So we're developing this case for how large artery stiffness is causing these dysfunctions in the brain. So now that I probably convinced you that getting old and is bad for your arteries, you're probably wondering, what should you be doing about it, right? So how can you go about keeping your arteries young? And unfortunately, I do not have any groundbreaking advice for you today. This is the same advice that you hear from everybody, but it's the same advice because it's true. The best things you can do for your arteries are exercise, particularly aerobic exercise, and eat a healthy diet. However, we do know that a lot of people aren't able to exercise or they choose not to exercise. And so in my lab, we're looking at, are there compounds like medications or supplements that we can give that would decrease the stiffness of the arteries and therefore improve those brain arteries and maybe reduce Alzheimer's disease risk. So one of those that we're looking at is a compound known as pyridoxamine. Don't write that down because you cannot get this anyway. You can't get it from your doctor, you can't get it from the grocery store. But pyridoxamine is a compound that's a form of vitamin B6, not the B6 that you get at the grocery store. It's a little bit different. And just by the little bit of difference that it has, makes it so that it reduces large artery stiffness, or actually prevents the stiffening of arteries with age. And so what we've been looking at is, can we give pyridoxamine to prevent the stiffness and thereby prevent all those bad things from happening in the brain? So we're still working on trials of this and we'll see what happens. And then we're looking at other compounds too. Before I summarize up all that I've talked about, there's one more thing I want to talk to you about. And that's an area that I think is really important and it's been being overlooked in a lot of studies, research studies, including things that I've been doing, admittedly. And it's something that we're trying to work on in the future. And that is that 2/3 of patients with Alzheimer's disease are women. And so there's a past history of doing a lot of studies just on men. And here we have it, 2/3 of people with Alzheimer's disease are women though. So we really need to be thinking about how are men and women different? And so if you think about it, you can stop and think about this for a second. Why are women more likely to get Alzheimer's disease? You might want to say it's because women live longer. And the longer you live, the more chances you have of getting Alzheimer's disease, right? That's a common excuse for this pattern. But even if you account for the longer life expectancy of women, they still have a higher rate of Alzheimer's disease. So really think there's something different about men and women that's making women more likely to get this disease. And one of the obvious causes would be the differences in sex hormones. And that women go through menopause, around the age of 50. And during this menopause, they have this decline in estrogen. So estrogen is really this protective molecule for the vasculature and also for the brain. And so this decline in estrogen could lead to an increase in this damage to the blood vessels in the brain. So we're really interested in figuring this out and how that damage to the blood vessels could be affecting this increased risk to Alzheimer's disease. And one of the other things that happens post-menopause in women is there seems to be a fuel switch in the brain. So think about this as if you had your brain, just like this picture showed, like attached to a cord and you're plugging it into the wall. So you plug it into an outlet in the wall. And when you're young, that outlet is supplied by, say, solar energy. Then after a woman goes through menopause, pull that plug out, plug it into a different outlet. The outlet's still giving the same amount of energy for that brain to work, but now that energy is coming from, say, nuclear power. And so we think that there's something about this switch that's happening with menopause in women in their fuel usage in their brain that we're trying to figure out, is that related to their increased risk with Alzheimer's disease? And is it actually happening in the arteries too? And so these are some of the concepts that we're really trying to work on now. So to summarize what I've shown you today is that I've shown you that aging is or leads to an increase in stiffness of the large arteries. And we talked about that being like pitching the skin on your hands. We lose that elasticity in our skin. We also lose it in our arteries. And then we talked about how that could be damaging to those brain arteries, such as having that greater pulse pressure in the brain could be damaging with the balloon and straw example. And now we're really trying to make the connection with both mouse and human studies that this increase in damage to the arteries in the brain is now leading to Alzheimer's disease. And so the question is, what could you do to stop it? And so all we know right now is that you can exercise and you can have a healthy diet, but one day maybe we're going to find a drug to target these pathways. And so to go back to that metaphor that I started with at the very beginning, where I was talking about Alzheimer's disease being about plaques and tangles, and how those were like having those trees falling on the telephone lines and the fibers within the telephone lines becoming frayed or tangled. I like to think about these changes to arteries with age are the storm. So this storm has come in. These arteries have changed, and this storm has caused these trees to fall over. And so we see that tree fell over, and that's a really obvious sign. And we're like, well, that's the cause, obviously, of those telephone wires not working anymore. But what if that storm is doing something different? It could be having damage at different parts in the wires, and these are things we need to figure out. Or that storm brought in that hail, which was like those free radicals, that are also causing damage somewhere where we're not seeing it. So my goal here is to, rather than trying to stop the trees, is to really think about stopping the storm. And so that's why I have my green arrow or my green stopping point on the slide. We're going to stop the large artery stiffness, rather than we're going to try to do it down at the Alzheimer's disease, which maybe is a point too late. And that's why a lot of those drugs aren't working. So that wraps up my presentation for today. First of all, I would obviously like to acknowledge all the people who helped with these projects. So my lab, which I called out already, Grant Henson and Nick Winder, who did a lot of work on the blood flow work, as well as Emily Reeve, another graduate student who is really handling our animal models right now. But I have a whole group of undergraduate students and graduate students. And I even saw a former undergraduate student is in the audience. So working on these projects. A number of collaborators also, particularly those people at OHSU who are really helping with those techniques that we can't do here, or they're helping us get new techniques set up in our lab. And then lastly, the funding. There's a list of funding here, but I have 3 of them that I want to call out to you. So the first one that I want to point out is this, actually second from the bottom, which is the Oregon Charitable Checkoff Donation for Alzheimer's Research, which is a big, long name. But this is a research fund that is administered by OHSU. And what it is when you do your income tax return for the state of Oregon, you have, if you're getting a return, like they're giving you money back, they give you like a whole big list of things that you can check off where you would rather donate your money to rather than have them give the money back to you. And Alzheimer's disease research is one of those things that you can check off. And so we got a grant from there. And that blood flow study that I showed you was actually funded entirely by that grant. So it came from Oregon taxpayers essentially to fund that research. We also have funding from the Alzheimer's Association, which is the biggest association within the US for supporting Alzheimer's disease, both research, as well as they give a lot of advice to patients and caregivers. And so you see things like Walk for Alzheimer's Disease and different ways you can get involved in that organization. And finally, the John Lewis Family Fund is the one who's funding us in these new studies with women in menopause and the effects that are happening in the brain, as well as we're working in collaboration with people at OHSU for those studies. So really thankful for those donations as well. And with that, I will open it up for any questions.
Thank you, Dr. Walker. That was a fascinating presentation. And I personally find it very interesting. My grandfather passed away of Alzheimer's. And I know that it seems like we're being able to diagnose or getting kind of back to your first poll, we're getting close to being able to determine before death whether or not it's actually Alzheimer's, because I know it's been a number of years now, but I know it was, well, we don't really know. We can't tell, whether or not it's just normal aging, a little bit of senility coming in, or if it's something further. So we do have some questions that are showing up in the chat, so I'm going to read those off. If everybody else has some, please feel free to put them in there and we'll just kind of run through them. So the first one, can stiff arteries be made more supple? So once they're stiff, do you have any recourse?
Yeah, so I mean, there have been some compounds that have worked in mice that they were able to actually reverse some of the stiffness. So they target some of the things, not just the elastic proteins, but there's other things where proteins get stuck together and make your arteries stiff. And they were able to target those, and it worked really well in mice, and it was going to be amazing. And then it didn't work in humans, just as all of these things have been working. So there is a question of like, maybe there is a way to do this. We haven't found it yet, though.
Okay. And next question was, are stiff arteries genetic?
That's a great question. So your genetics plays into this a little bit. Like some parts of, a little bit of your blood pressure is genetic, right? And blood pressure can affect the stiffening of the arteries. But a lot of it is just, it happens. It happens over time. So the best ways to avoid it are things like exercise and diet. But there's no like obvious genetic cause to large artery stiffness. Nobody's found like 1 or 2 genes that are really predictive of having stiffer arteries. So, there might be a few things that are coming into play with genetics, but nothing obvious.
Sorry, I had actually heard that there was a hereditary component too, but sounds like that's not necessarily true, other than hereditary in terms of your fitness. Next question, could women be diagnosed at a higher rate because of cultural factors?
That's a really interesting question. And so there are definitely cultural things that go into diagnosis. I don't know exactly where the questioner is thinking about this, but there are things like women go to their doctors more, which might make them more likely to be diagnosed. And of course, women living longer is another piece of it. There's also with diagnosis of Alzheimer's disease, especially, as you mentioned, we're getting better at doing it with the brain imaging. But if you're just looking at people's cognitive decline, a lot of that depends on where you started before you started the decline. And so there's findings in women have, different parts of their brain, there are different types of memories that they're better at keeping than men. And so that maybe their decline is different than men. So yeah, there could be cultural factors that play into this for the women versus men. I mean, a big thing also, which we can't study in mice, obviously, but a big thing that, we've been thinking about as far as risk factors are other cultural things such as, race and, what is structural racism doing to people? It's increasing their stress, which increases their blood pressure, which increases all of these disease indices. And so there are factors in that way that could influence this. I don't know, particularly for men versus women, though, what exactly the cultural factor would be.
So what are the best things that we can do in terms of lifestyle to keep our brains healthy as we age?
Great question. So obviously I told you exercise, healthy diet. The other thing is doing an assortment of brain activities. So if you do just one sort of brain activity, you get really good at doing that sort of brain activity. And that's not necessarily the best thing for aging. So if you do crossword puzzles every day, I mean, that's great and it's fun and you should keep doing your crossword puzzles, but only doing crossword puzzles isn't the best mental exercises. So you want to do a lot of different variety of exercises for your brain as you get older. We also know one of the best things for aging is to maintain social involvement, which is a horrible thing to say right now because this is a big problem with COVID, right? But to have that, fulfillment, to maintain social interactions with people, because that's using parts of your brain as well, is really important to keeping your brain healthy as you get older.
Okay, what is the difference between Alzheimer's disease and dementia?
Oh, good question. So dementia is an umbrella term. So dementia includes a bunch of different things that lead to having impairments with memory, essentially. And so Alzheimer's disease is one of those. And there's other ones. There's Lewy body dementia, there's frontotemporal lobe dementia, there's a new syndrome that's called late that they actually realized that people who are being diagnosed with Alzheimer's disease at really old ages, like say 90 or above, actually probably have the syndrome known as late and not Alzheimer's disease. It's kind of a fitting name that they called it late, but it has a really long name as well. So dementia is the umbrella term. And then there's all these things that fall under the umbrella. And when I teach this in class, I always tell people, and I bring up all those slides with the other damage in the brain and, how much percent is, and I say, under this umbrella, they try to diagnose people, but there's so much crosstalk between that. And there's also vascular dementia as a type of dementia as well. So there's so much crosstalk between all the different dementias that, you could have more than one thing going wrong with your brain.
All right, Chris Minson and what is this, Human Physiology Lab believes hot exercise can improve arterial flexibility. What are your thoughts on this?
I think Chris is in the audience, so we should just ask him instead.
I would say this, when I read it, I thought this sounds like a setup to me.
I know. Did you ask somebody to ask this question? Yeah, so Chris Minson's group has a lot of great studies going on that show that heat therapy, so like sitting in a hot tub or sitting in a sauna, has benefits for the vasculature. And one of these things could be improving the stiffness of the arteries or preventing that increase in stiffness as we get older. And so that's another avenue. And maybe that's another way in case you can't do exercise. The thinking is the alternative might be to sit in that hot tub. And that it might be having a lot of the similar effects. It's sort of like a heat stress on the body, similar to what you get when you exercise. So I probably said that wrong and Chris can correct me later.
Okay, we'll move on from that. Have you noticed any correlation from patients who have undergone open heart surgery and Alzheimer's diagnosis soon after?
That is a really interesting question, and I don't know the answer. That's a little bit too clinical for me. I'm sorry, I stick on the basic science research part of that, but it's a good, interesting question that maybe there's some literature on. If we did like a PubMed search, we could find something about it.
All right, and then then the next question was the same question as above, but with regard to strokes. So
I do know, I can do the strokes question.
Okay.
I do have strokes. So strokes, because they are like, those are the blood vessels in the brain. So you can have two different types of strokes. Either there's a clot in your brain that's stopping blood from getting to your brain because of that, or there is a bleed in your brain. But in any case, it means the blood isn't getting to the brain where it needs to be, right? So you're not delivering those oxygen, the glucose, all those other things. And so the brain tissue is getting deprived of oxygen and it could die or other things could happen. So there is a form of dementia known as vascular dementia that means that you had a stroke and you had dementia form from that. But there are people doing research to show that it actually is related to Alzheimer's disease as well. And so we think it's because when you have that loss of blood flow to that section of the brain for a while, even if you get blood flow back, or especially actually when you start getting the blood flow back to that area, you get an increase in that free radicals and an increase in inflammation. And that inflammation can trigger Alzheimer's disease. And so I'm actually collaborating with somebody up at OHSU who does really cool studies on stroke and Alzheimer's disease and how they're connected.
Okay. So our next question says, I heard that trying to use your left hand, well, this would be for people who are not left-handed. I heard trying to use your left hand to do similar tasks that you do with your right hand can help stimulate other parts of the brain to help as a prevention method. What are our thoughts on that?
What are my thoughts on that? I mean, it sounds like it might work, but I would like to see the clinical trial on that. And as far as I know, nobody has proven that with a study, but possibly could be something. But I'm a scientist, so I want to see the hard data, the results from that study before I start trying to do things with my left hand, because I'm pretty awful with my left hand.
We're going to talk about puzzles and such for people to do. And I've heard the thing about the crossword puzzles before. Are there particular types? Is it like word puzzles? Is it anything in particular that helps?
I mean, I think it's a variety, right? You don't want to just be doing math. You don't want to just be doing words. You want to be doing sort of everything. You want to be reading, you want to be writing, you want to be doing a puzzle, you know, probably physically doing a puzzle probably helps, you know, putting down the screen and doing something with your hands. So yeah, just a variety of tasks is really what you're aiming for.
Okay. Let's see, association between exposure to general versus regional anesthesia. What the heck is this? Let's see here.
We got linked to an article.
We've got yes, there's a link. Yeah, a link to the journal online there. So I'm not sure if that was a question or just sharing the link in the article there.
Interesting. I'll look at the study.
Yeah, I was going to say that's a little bit of a deeper dive there. So let's see. Are we looking for one silver bullet at the expense of solving many similar issues? Or sorry, not similar, smaller issues. Are we looking for one silver bullet at the expense of solving many smaller issues?
That's a good question. And I think that I agree. I mean, I put it as I'm trying to prevent the stiffening of the arteries, but this is me. This is what I do, right? I study artery stiffness and I study aging and vasculature, but there's probably other things we should be doing too. And so Alzheimer's disease is caused, especially if we're talking about late onset Alzheimer's disease, like people who are getting Alzheimer's disease after the age of 65, it's a multifactorial disease, meaning there's multiple things that are coming into play. So it's that you're old, you have bad arteries, maybe you have high cholesterol, you have high blood pressure. It's a lot of different things that came into play probably that caused this disease. And so we probably should be trying to fix all those little things at the same time. And so I agree, there probably isn't a silver bullet to just solve this. And maybe it is a bunch of little things to do.
Do we see extremely healthy people get Alzheimer's? So those people that have been running like marathoners, what have you, that have been healthy their entire life, don't smoke, don't drink, don't do any of that, and then they still get Alzheimer's?
Yes. Yeah. So there isn't really an obvious, this person escaped. So we know that doing those healthy things make you less likely to get it, but people still get it even if they're doing all those healthy things. And so, maybe there is a genetic component to it. There are genetics involved in Alzheimer's disease. Before I was talking about arterial stiffness, which there isn't as much genetics involved in that, but with Alzheimer's disease, there is. So if you get Alzheimer's disease when you're younger, so before the age of 65, it's probably genetic. And we know the genes and the gene mutations that lead to Alzheimer's disease when somebody's young. There's people who are getting it when they're 40 or 50. And then if you get Alzheimer's disease when you're over the age of 65, so late onset, it's less likely to be genetic, but there is a gene, the ApoE gene, that if you're a carrier of the type 4 of this, so 2 ApoE4 genes, that you're more likely to get Alzheimer's disease. And, if you have 2 of those, you're really likely to get Alzheimer's disease. So those are probably the people that are getting it, even though they're doing all of those good things, they still have a genetic risk factor. It's not a guarantee that they're getting it, but it's making it a lot higher risk.
Okay. You mentioned that the plaques and tangles are protein. Is it our own genetic protein or something else? Are those protein cells connected to our vascularity?
Yes, so those are proteins. There are proteins that are made by the cells in your brain, primarily by your neurons. And so tau, the one that makes the tangles, is a protein that's actually a good protein. So it's part of that, the wire of your neuron. And it's important for neurons to work well, but it becomes phosphorylated, which is just a way that it changes when it's damaged. And then when it's phosphorylated, it clumps together. And so it forms aggregates. Amyloid, or those plaques, are caused by amyloid. And amyloid is a protein also produced by your neurons, but maybe some other cells in your brain. And amyloid is a really interesting one because it's produced by a precursor protein. So it starts off as a big protein and then it gets cut up into pieces. And how it gets cut up into these pieces actually determines whether or not it clumps together. And so there are forms of where it gets cut up that are more likely to clump versus less likely to clump. And those could potentially change with age or how much inflammation you have in your body. And genetics as well.
We have a question here that's interesting. It says, how does exposure to metals affect Alzheimer's risk factors?
Yeah, so that was sort of a hot topic years ago.
Oh, wow. Mm-hmm.
Yeah. There's not really a lot of strong evidence for people who are having normal exposures to metals, like, every day, whatever it is, using your deodorant, those type of things. There isn't evidence that that is causing an increase in Alzheimer's disease. People who are industrially being exposed to these things, maybe, but not for your everyday life.
Well, that's good to know. That's a bit of a relief. So we got 1 minute left. I'm going to let Ellie ask the last question because I believe that she had one. And then we can wrap up with our winners.
Hi, I do have one last question. So this is just kind of tying stuff into COVID right now. So I know that the type of pneumonia that's often associated with COVID-19 can cause a lot of long-standing damage to the tiny air sacs in the lungs. And so then long-term breathing problems result, which then obviously is issues getting oxygen to the blood. So I'm just kind of wondering, in your opinion, if you think that those that are recovering from COVID may inadvertently be more susceptible for Alzheimer's later on due to artery health. Obviously, there's no way for you to know fully, but I'm just kind of wondering your opinion on that.
The short answer is I don't know, and it'll be interesting to see what happens. I think with COVID, the oxygen getting into the blood is a big piece of it, but the other piece of COVID is that we're seeing people actually have direct vascular damage. So there's things like they are getting more platelet aggregation, blood clots, strokes. People are getting who are young are getting strokes from this. And so the thought is that there's actually, changes to the cells in the arteries with COVID. And so we don't know what that is doing either. That could be directly, it could be those, mini strokes that we're not seeing, or it could be some other changes to the arteries that could be happening. So there's just a lot we don't know right now.
Yeah, it's super complicated. It's just such an interesting thing to think about for future use. Okay, well, I just want to thank you, huge thank you for presenting. Huge thank you to the PDX Ducks board and to the Alumni Association members in the audience. So we have numerous events happening throughout the year. So just make sure that you're checking your email and your website for some of the virtual ones that we will be doing. And now I will announce the two winners. If the winners can just wait at the end so I can just verify that we have the right email on file to send you your gift card. So the first winner is Pat Sullivan. It might be Patty, but that is winner number 1. And I think that person is on. And then
I'm here.
Yeah. Okay, there Okay, and then the second winner will be Kathleen Hanna, who I also think was on. Is Kathleen Hanna?
Yep, I see her on our list. She's here.
Okay, awesome. Cool. Well, thank you to everyone. And if the two winners can just wait a couple of minutes after, have a great evening. Thank you again.